So to close the session, I want to tell you a little bit about what's going on in the cancer research community. So until about 10 years ago, we sort of had two separate communities. One, cancer biologists trying to decipher the molecular underpinnings of what was causing normal cells to turn into cancer cells, and another community of drug developers, which for the most part were relying on fairly empirical strategies to see what kind of compounds would kill cancer cells and then see if we could give them safely to patients. But about 10 years ago, the field has undergone what I consider a really remarkable renaissance, where the lessons from the first community can be directly applied in the second community. So we now have this era of what we call targeted cancer therapies. And so these, it's kind of a loosely used term, but it is meant to refer to drugs that are developed specifically to attack a molecular lesion in a cancer cell and therefore eliminate a lot of the side effects of chemotherapy and really go after what we call the driving mutations in the cancer. So as you might expect, the excitement of this has been transforming for the field, but these drugs are not cures on their own. And so I want to tell you about where we stand in terms of trying to overcome the resistance that can develop to these new therapies. And I want to introduce you to this by saying that there's a lot of lessons that our field is now learning from colleagues in infectious disease who have learned this over decades. So as you all know, single drug treatments for most infections can fail because we select for cells, bacteria, or viruses that develop mutations to the drug target. And organisms like HIV, which have incredibly high mutation rates and replication rates, are notorious for not responding to single agents only for a period of months before developing resistance. But yet, by understanding each of those successes and then rapid failures in a precise way, combinations of rationally designed combinations have been developed that can now either control or eliminate infections. So resistance to chemotherapy drugs, which have been used for decades, is actually still today rather poorly understood. But this new class of compounds, these molecularly targeted therapies, we've actually made tremendous progress over just a short period of time in understanding how resistance develops. And the largest body of knowledge is around these classes of compounds called kinase inhibitors. So many of the driver mutations in cancer are in genes that encode kinase enzymes, and it's been possible now with great success to develop fairly specific inhibitors of many different kinases. They work by, in general, competing for ATP at the ATP binding site, and have caused tremendous response rates even as single drugs in many different cancers. But in the examples in which resistance has now been worked out, these include leukemias, sarcoma, lung cancer, and a growing list. The most common mechanism of this acquired resistance is due to mutations in the kinase target that circumvent the binding site in some way, yet retain the enzymatic activity of the kinase so that the tumor can continue to grow, but the drug can get around it. But that disappointment has actually been rapidly overcome in a number of examples by developing second-generation compounds that can now work against some of these resistant mutants. So in the same way that combination antibiotics or antivirals have been deployed, a similar strategy is starting to be deployed in cancer. What I want to tell you about now, though, is a story that involves prostate cancer, in which we don't have an example of a driver kinase mutation, despite lots of hard looking for such alterations. And I want to disclose that I'm going to discuss a drug that I stand to benefit from personally if it happens to be approved. So to set the stage, the treatment of prostate cancer in men who relapse after surgery or radiation therapy for decades has depended upon removing testosterone from the system. So prostate cells normally and prostate cancer cells depend on testosterone or its another form, dihydrotestosterone, as a steroid hormone that the cells need to grow and survive. And so the main treatment for prostate cancer, developed several decades ago and still used today, is to lower serum testosterone levels. That works effectively for a period of time, generally a couple of years, before the tumor becomes resistant. There's another class of compounds known as anti-androgens, which in a sort of very parallel way to the kinase inhibitor story, block the active, the ligand binding site of the receptor. The leading drug today is one called bicalutamide. And it's an effective drug when used early in the disease, but again, resistance develops quickly. But that drug, as well as an earlier version of it, have liabilities, one of which is that they both, although they were designed and have properties as antagonists or inhibitors, they acquire with time in patients the sort of paradoxical phenomenon of actually stimulating the androgen receptor. So I'll illustrate that in a very simple cartoon. So if we picture the progression of the patient's tumor as this rising line, we institute hormone therapy. Responses are quite dramatic in most men and last for a period of time. The disease recurs, and the first maneuver that the clinician takes when seeing this recurrence is to stop the anti-androgen, typically bicalutamide, and about 20-30% of the time, the patient will actually benefit from stopping that drug, sort of illustrating quite clearly that the drug has mixed antagonist-agonist properties. That maneuver can work for just a very short period of time, and the tumor outsmarts it. So this phase of the disease for years has been called hormone refractory or androgen or testosterone-independent prostate cancer. And it's presumed that a whole new set of lesions is developing, and the question that we asked many years ago was, what are those new lesions? So to do that, it's very difficult to analyze these tumors directly from patients, so we took advantage of some model systems that we and others have developed over the years in which we can grow prostate cancer cells from human tumors in mice in this phase here when the mice still have androgen present. We can actually castrate the male mice, and the tumors will shrink and then undergo this same progression phenomenon. So what my group did was take paired samples of tumors at this stage and this stage, and used genomic technologies such as transcriptome profiling, where we can look at all the RNA messages in the cell and analyze a number of these tumors and tried to understand what was different as the tumors progressed. And what we found we found a number of changes, but what sort of stood out and rose out from all of the data was that the androgen receptor itself, the target of testosterone, was more highly expressed in the castration-resistant phase, consistently across all the models, and as we and others have subsequently shown, also in patient samples. We then did the typical experiments that a biologist will do in this case, and that is ask if this is just an association or is it causal, and we proved that it was causal, at least in these model systems, by taking the hormone-sensitive tumors, overexpressing the androgen receptor, and that was sufficient to make them now grow in a castrate animal. And the reciprocal was also true. If we inhibited the androgen receptor using a technology called RNA interference, which is not a way to make a drug yet, we could restore their dependence on hormone. And also importantly, this same maneuver was sufficient to cause the drug bicalutamide to now undergo that conversion from an antagonist to an agonist. So this, to me, set a very clear path as to how a next-generation drug might be searched for, and that would be to screen compounds against prostate cancer cells in which we had engineered this higher level of androgen receptor expression. And I proposed this back 6 or 7 years ago to many drug developers and couldn't get any uptake in the pharmaceutical industry for a number of reasons, one of which was this idea that the disease was not felt to still require the androgen receptor in this late stage. And although we had shown it in a model system, it wasn't clear that that was going to be true in patients. So I decided to partner with a colleague, Michael Jung, a synthetic chemist at UCLA, to see if we could tackle this problem in the academic setting. And what we did was take advantage of a lot of chemistry that had already been done in the field of steroid hormone receptors, and in particular take advantage of this compound that had been described in a Roussel-Uclaf patent about 20 years ago that caught Mike's attention because it has this very high binding affinity for the androgen receptor. This is about 2 orders of magnitude tighter than the current clinical drug bicalutamide. We quickly found out why this had gone nowhere, because when Samdi Ook and Mike's group made the compound and we tested it in our system, we found that it was a potent stimulator of the androgen receptor and therefore not of any clinical use. But by making changes in the molecule we were able to build in properties of antagonism. And the main change that led to the compound that I'm going to tell you about was to remove this group over here and put bulky residues that cause the molecule to now display properties of inhibiting the receptor rather than stimulating it. We don't know exactly what is happening at the atomic level because we haven't solved the crystal structure of the compound bound to the androgen receptor, but lessons from breast cancer and the estrogen receptor suggest that what's happening is there's a helix that when normal hormone binds, folds over on top of it and locks it in the ligand binding pocket, and these bulky residues are believed to block the folding of that helix. So the compound that we liked the most, we called RD162, was licensed by this company, Medivation. And they licensed it based on this kind of data that we generated in the lab, in which we showed that the compound could shrink tumors in mice that were castrate-resistant tumors. So what's shown here is what is known in our world as a waterfall plot, and each one of these bars represents a mouse with a tumor. The 0 line represents if the size of the tumor does not change over a 28-day treatment period, the bar will be right at 0. So you can see that the yellow ones, most of the tumors grew. With the current drug, most of them also grew, but with the new drug, all the tumors are shrinking in the mice, and some of them are shrinking completely. So this is why the company was interested in taking it forward. In the meantime, we asked, why is it really working so much better in these mouse models? Is it because it has a higher binding affinity? I mean, it definitely does, although when we built in those other properties to make it an antagonist, we lost some of the affinity. What we actually found out is that it's when it binds to the receptor, it causes some completely different properties or confers different properties upon the androgen receptor. So normally the androgen receptor is located in the cytoplasm of the cell until a ligand such as testosterone binds to it. And when testosterone binds, it undergoes a conformational change and translocates to the nucleus and binds to target sites in DNA and turns on the transcription of a whole set of target genes. There are roughly 20,000 binding sites in the genome that we can identify under this context. The current drug, bicalutamide, actually doesn't differ that much in its effects on the androgen receptor. When it binds, it also moves to the nucleus and binds almost the same number of target sites. But when we look at the effects of the new compound on the androgen receptor, we see two differences. First of all, only about a third of the pool of androgen receptor in the cytoplasm is able to make it across to the nucleus. And of that pool, we can find no evidence for DNA binding at any of the classic sites. So we think this has a very different property, and if we could get a better handle on how it's binding at the atomic level, we could perhaps even do better. In the meantime, the company Medivation decided to take the drug forward into a phase 1 clinical trial, which was run at Sloan Kettering by my colleague Howard Shear. And as with all first-in-human drug trials, there's a period in the early phases in which a very limited number of patients are given doses that are believed to be subtherapeutic in order to look for any unexpected toxicities. So this is called a dose escalation trial. Usually 3 patients are treated at a time, watched for a month, and then we go to the next dose. So the first dose level was 30 milligrams, and the first 3 men who were treated, and these were all men who had failed standard hormone therapies for prostate cancer and essentially exhausted their typical treatment options. Remarkably, all had a subtle decline in their level of prostate-specific antigen, a tumor marker in the blood of prostate cancer patients. The next 3 men at 60 milligrams also had that happen. So this was obviously an extremely exciting result because it was at least indicating hints of anti-tumor activity. So the company decided to amend the study and now aggressively enroll a larger number of patients at each dose level once the safety had been established in the first 3 patients. So a typical phase 1 study is usually about 30 or 40 patients. This quickly grew to 140 men who were treated at these 5 centers over about a year and a half. And the very exciting results from that trial are shown in this now waterfall plot of human patients. Here the 0 line is here, and then this red dotted line represents a 50% decrease in the level of serum PSA in the blood in these men. And you can see that roughly 60% of the men who had not been treated with chemotherapy but had failed other options had this kind of response, and about 50% of those who had failed chemotherapy. So this is a very exciting result for a new drug in prostate cancer, and has led the company to now do a formal randomized trial comparing this drug to standard treatments of supportive care in this population, and the results of that trial are expected to read out later this year. In the meantime, there's this other group of men who are not responding to this compound at all. If I drew those red circles a little bit wider to include these patients, I could tell you that those men respond very briefly and then the disease progresses. So I think we clearly have an example of a drug that's working in a subset, a large subset, fortunately, of men with the disease, but we'd like to understand why it's not working in these other men. The tumors express the androgen receptor, we know that, so what's going on? Well, it's very difficult to obtain biopsies from the tumors of these patients, although we're gradually collecting them. So in the meantime, we've turned to mouse models again. And we're taking a guess. In this case, we're guessing that it might be due to mutations in a gene called PTEN. So PTEN is a tumor suppressor gene that's involved in many different cancers. In this case, loss of this gene causes cancer because it turns on, it activates various pathways that PTEN normally regulates, including one called PI3 kinase. So why do we pick PTEN? Well, it's common in prostate cancer. It's about 40% of men with prostate cancer will have deletions of PTEN, and that roughly matches the frequency of resistance to the drug. In addition, there's evidence from breast cancer where PTEN and alterations in the pathway that it regulates also occur, that resistance to hormone therapy in breast cancer might be linked to this pathway. And finally, and perhaps most practically, we could test it pretty quickly because we have a very nice mouse model in which the PTEN gene has been specifically deleted in the prostate gland, and those mice develop prostate cancers, which we can visualize by MRI scans made for mice. And when we look at the effects of the compound in that drug, of the compound in that mouse model, again, this is now a waterfall plot again. These are 5 PTEN mice whose tumors shrank just 20% when we give that compound, as opposed to another mouse model in which the tumors shrink almost 100% when given the compound. So this is consistent with the idea that perhaps PTEN loss is a resistance marker for the drug. Now, of course, a negative result like that always has to be followed up with other questions as to why was the result negative. Did we actually get the drug effectively into the tumor cells? We think we did because we're seeing that the expression of 3 different genes that are regulated by the androgen receptor is down by about 40-fold in these tumors. So that's a pretty convincing result. And we can also see that the androgen receptor is no longer purely in the nucleus anymore when we treat the animals with this drug. So it's actually doing what it's supposed to do to the androgen receptor. So why aren't the cells dying if we're inhibiting the androgen receptor? Well, it's possible that this PI3 kinase pathway, which is turned on when the tumors in the tumors that lose PTEN, which we know regulates the survival of many cells, is still keeping the cells alive. So what we've now done is inhibited both pathways. And what's come out of this is we learned that when we studied what happened to the tumors in which we inhibited the androgen receptor, what we found is that paradoxically, we gave our androgen receptor drug and turned up the activation of the PI3 kinase pathway. And the vice versa was also true. When we added a PI3 kinase pathway inhibitor, we activated the androgen receptor. So those surprising findings have led us to elucidate the fact that there's these previously unrecognized negative feedback loops where one pathway, the PI3 kinase pathway, regulates the androgen receptor pathway, and vice versa, the androgen receptor pathway activates the PI3 kinase pathway. So these signaling networks, which we had in our field have typically been drawing as linear diagrams, are actually clearly much more complex, but it does, and there's a lot more to understand there, but it begs the question, what would happen if you gave these 2 drugs together? And the answer, for the first time in this PTEN model, is we see dramatic tumor regressions. This is 4 mice in which the tumors are decreasing to 80 to 90% of their pre-treatment volume. And when we actually look at the tumors in the animals, it's mostly scar tissue at that point. We also see it in another model as well. So what I've tried to tell you is that first of all, in this particular prostate cancer example, we have now a new set of drugs which are moving forward in the clinic. The one I showed you is called MDV-3100 that have clear clinical activity in men with late-stage prostate cancer. And we found them by looking at why they were failing the first forms of therapy, and the reason is by restoration of the androgen receptor signaling pathway. The second point is these PI3 kinase pathway inhibitors are making their way into the clinic pretty quickly. They're going to be tested in prostate cancer soon, and our data about those negative feedback loops is likely, it makes the prediction that in fact not only will they fail, they're going to actually cause the serum PSA level to rise in patients rather than fall, and that's going to cause massive confusion in the early days of those trials. So we are trying to get this message out to the clinical trial community to be aware of this. And then finally, we want to very aggressively encourage the companies that are developing these two compounds to work together and use a combination therapy. But I think the last point I want to make is that we're now, as we dig deeper into the mechanisms by which these targeted therapies work and then fail to work, we're understanding that the number one cause of resistance is mutation of the target, but the number two cause of resistance could be these previously unappreciated negative feedback loops that allow the cell to compensate for inhibiting one pathway by turning on another. But we have the technology to look for those quickly as we develop these drugs, and therefore come at the tumor with the second compound in a very rational way. I'd like to close with just listing several of the people in my group who've contributed to the project I discussed, and thank you for your attention as the last speaker in this session. Thank you.